right, can everyone hear me? Yes, they can. Great, awesome. Thank you for coming to this week's seminar. It's my pleasure to introduce Dr. Susan Racette, who's a professor in the College of Health Solutions downtown in Phoenix here at ASU. Susan, we're lucky enough that Susan joined us at ASU in 2022. Prior to that, she was a professor at WashU in St. Louis at the School of Medicine there. Susan studies how diet and exercise affect metabolism, cardiovascular health, cognitive health, and aging. She's really known for co-leading some work, which she'll tell us about today, which is the CALERIE trial, which is the first randomized controlled trial of of long-term calorie-restricted feeding to understand impacts on health span, aging, and disease. And currently, she explores a lot of research related to, time-restricted eating, exercise training, cardiac rehabilitation, and extending the health span. And before I say anything more that might scoop what she's going to tell us, I'll turn over. So thank you, Susan.
Okay, great. Thank you very much, and thank you for attending. It's always nice to have people in person, and hello to those on Zoom. So my background is in nutrition and exercise physiology. So my PhD is in Nutritional Biology from University of Chicago, and then I went to WashU School of Medicine in St. Louis to do my postdoctoral work in exercise physiology. So all of my focus is on nutrition, exercise, or the combination, and really for cardiovascular health promotion, healthy aging, and really health span, which I'll be telling you more about. So what I'm going to focus on today is calorie restriction, and this is not what we think of in terms of obesity treatment. I did actually start by doing obesity treatment studies, but this is calorie restriction for anti-aging. And so it's a little bit different than some of you might be familiar with, but hopefully I'll convince you that this is a worthy pursuit scientifically. And so let me begin. So disclosures, I have some consulting and then grant funding. They don't conflict with my research, though. And so I want to talk about two types of aging. So aging can be thought of in terms of primary aging and secondary aging. And primary aging is the progressive deterioration in tissue structure and function due to age. And this is considered inevitable, and this determines maximal lifespan. In contrast, secondary aging, which is also a deterioration in tissue structure and function, but this is due to things like diseases, adverse lifestyle behaviors, harmful environmental factors, and this is considered to be not inevitable, and that this determines mean lifespan, so the average duration at which someone lives. And so we know that there are many strategies that can impact secondary aging, things like not smoking and maintaining a healthy body weight and eating a healthy diet and exercising and all of those things. But what we really want to understand is, can we impact primary aging? So the thought that you can modulate aging with nutrition actually dates back a very long time. And Benjamin Franklin eloquently stated, to lengthen thy life, lessen thy meals. And so this concept of restricting calories dates back quite a long time. And so interestingly, to date, calorie restriction is the most robust intervention to delay aging. And I'll show you evidence for that. So there have been many studies in many different organisms that shows that calorie restriction extends lifespan. So first let me define calorie restriction. This is a sustained reduction in calorie intake with adequate intake of essential nutrients. And so that's important. We don't want to put people in a nutrient insufficiency or even deficiency. And so, but this is a sustained, not a short term, not a 12-week or a 6-month even. This is really a sustained reduction in calorie intake relative to baseline needs. And so it was shown in many lower organisms that CR extends both mean and maximal lifespan. And this also has been shown in organisms like dogs and some of the non-human primate studies with rhesus monkeys. And the studies really date back to around the 1930s when Dr. Clive McKay at Cornell University wanted to see if essentially growth retardation would lengthen the life of rats. And so what he showed, what this shows is percent still alive on the y-axis and age on the x-axis. And what you can see is that compared to rats that were fed ad libitum, rats that were calorie restricted by 30%, this is a 30% reduction relative to energy needs, had an increase in both mean lifespan and maximal lifespan. These were followed up by studies many years later by Dr. John Hollisey at WashU School of Medicine in St. Louis. He was my mentor for my postdoc. And he was also, so he was a physician who was not trained as a researcher, but was doing research long before many other physicians were doing research, and was also a real exercise advocate, but was also interested in nutrition. So he was doing studies, again with rats, looking not only at caloric restriction, but also with exercise. And so this also shows, similarly, the survival curves here relative to age for 4 groups. And these 4 groups were a control group, an exercise group, which you can see had an increase in mean lifespan, but not maximal. So the curve is shifted over, but the maximal did not differ from control. And then there were 2 CR groups, a calorie-restricted group and a calorie restriction with exercise. And so what this shows is that both mean and maximal lifespan were increased by CR, and that exercise really didn't seem to have an added benefit. So this is suggesting that CR is acting through some way that's distinct from exercise. Now, what we're really interested in for humans is not just lifespan, but health span. And so health span can be defined as the number of years lived in good health with high physical and cognitive function and a high quality of life. So you hear about people that, live to whatever age, but are they still, living independently? Are they still able to do the things that they enjoy in life and spend time with the people that they want to spend time with? Or do they really have a lot of disability that impacts their quality of life significantly? So we really want to increase health span, and it's been stated that there's about 12 years difference between health span and lifespan, meaning that at the point when people no longer have adequate health span, they still live 12 plus more years before they die. And we really want to close that gap and see what we can do to increase people's health span. And so there's compelling evidence from cross-sectional studies that CR can actually increase health span. And some of these studies were done by a colleague of mine when he was at WashU, Luigi Fontana. And he's shown here. So he had a very unique study of people who were in this, what was called Crohn's Society, calorie restriction with optimal nutrition. And what he had people fly in from all over the world, and he studied them. And so these are people who voluntarily had been following a calorically restricted diet on their own, not as part of a research study, for about 6 years. So long-term commitment. And what this shows is compared to controls, the CR group had better metrics on pretty much everything. So they were matched for age, but they had a lower body weight, lower body mass index, which is more in the healthy range. Their percent fat was drastically lower. And then all of these standard metrics, lipids, blood pressure, glucose, insulin, C-reactive protein, they were all more favorable in the people who had been following a calorically restricted diet. But you could say, well, they were just different at the outset. That doesn't really necessarily tell us that it was calorie restriction. Well, he had a group bring in their results, medical records, all kinds of things from before they started CR. And what you can see is they were actually very much like the controls at baseline for all of these different metrics. So they had pretty much the same kind of health and risk profile. So providing some evidence that there really is some merit to this calorie restriction. But of course, that wasn't a research study, but that set the stage for our study referred to as CALERIE. Yes, CALERIE is misspelled, that's an acronym. It stands for Comprehensive Assessment of Long-Term Effects of Reducing Intake of Energy. And so this was funded by the National Institute on Aging because they were very interested in this and increasing health span. And so the CALERIE studies have been ongoing since 2001, and it started out as phase 1, which I'll show you some data on. Those were 3 randomized controlled trials at 3 clinical CALERIE sites. This led to phase 2, CALERIE 2, which was a single randomized controlled trial. So then all the sites came together with a single protocol. And we're now doing the CALERIE Legacy study, which is an observational follow-up of the people in CALERIE 2. And so I'll show you more details about those. And so we're going to focus right now on Calorie 1. So the 3 clinical sites that were funded to do this study were Pennington Biomedical Research Center in Baton Rouge, Louisiana, Tufts University, their Jean Mayer USDA Human Nutrition Center on Aging, and WashU School of Medicine in St. Louis. And then Duke Clinical Research Institute was the coordinating center, and it was funded by NIA. And NIA officers were very much involved in the study throughout. And so this shows that we all had different designs at the 3 sites. The sample sizes were the same. There were more women at all sites, but the ages varied. We did a little bit of an older age group at WashU. We had the requirement was that everybody had to be non-obese, so they had to have a BMI less than 30, but we were studying predominantly people who were overweight but non-obese. And the level of calorie restriction varied from 25% at Pennington, 10 and 30% at Tufts, 20% at CR. And if you recall in the rat study that Clive McKay did, it was 30% restriction. And that's in the animal studies, it's usually a little more restrictive. And the duration of the interventions were 6 to 12 months. And so what did we find? Were there favorable results? So we found, kind of collaborating all of the different results or collating all of the results, there were beneficial effects on many things. Things you would expect: fat mass, both whole body as well as visceral and subcutaneous when you look regionally, improvements in various different hormones, insulin sensitivity improved, blood pressure, serum lipids and lipoproteins, left ventricular diastolic function as a cardiac measure, inflammatory markers like C-reactive protein, muscle mitochondrial gene expression, DNA damage, and aerobic capacity. So lots of favorable results on various different systems. Were there negative effects? Well, as you would expect with weight loss, there's lean body mass loss. And in absolute terms, lower body strength decreased and bone mineral density. So those are some of the things we need to be aware of. But essentially, these results then made it so that we concluded, okay, we're going to go on to phase 2. So we determined that CR is feasible for up to 12 months. It has many beneficial effects, and that a longer-term study is in fact warranted, which was the plan from the beginning. So then we went on to phase 2. And so phase 2 was designed as a 2-year intervention as opposed to 6-month to 1 year, 25% CR, and in 220 participants. And so it was based on various things that we observed in phase 1. And so the design shown here, we had very extensive screening. So what's really important is that eligibility is that people had to be healthy. They could not have elevated blood pressure. They could not have type 2 diabetes, cardiovascular disease. They couldn't have abnormal blood values on most things. They couldn't be on medications other than oral contraceptives. The age range was 21 to 47 for women, up to 50 for men. We were trying to avoid menopause. And then the BMI range between 22 to less than 28. So for reference, 25 is the cut point. From if you're less than 25, that's considered healthy weight. 25 and above is considered overweight. 30 and above is considered obese. And so we were studying people normal weight and mildly overweight. After screening and a long baseline assessment period of like 5+ weeks, they were randomized either to intervention or control. And this was a 2-to-1 randomization, so you can see the sample size is there, 2-to-1. 25% calorie restriction. So that means every day for 2 years, they were supposed to eat 25% fewer calories than they were at baseline. So that's a big commitment. That's not an easy thing, so keep that in mind as I show you some of the results. And so then again, we were doing assessments throughout the 2-year period. And one of the really exciting and important aspects of this study is we use the doubly labeled water method. And this is something that I did in my doctoral work, and that's just a wonderful tool. And so the important aspects of this is we wanted to prescribe their 25% reduction, their 25% CR accurately. And so the only way to do that is to know how many calories are they burning at baseline. And so doubly labeled water is the most accurate method for assessing total energy expenditure, non-invasively, so they lead their normal lives. They're not in a chamber. They're not tethered to a machine. And it's non-invasive, free living, and it's safe. And so we get accurate assessments. And we actually did this over 4 weeks. So we have 4 weeks of data for their daily energy expenditure over that period at baseline, upon which we could prescribe their 25% CR at the individual level. The other reason this is so useful is we can determine how adherent they are to the diet. So by measuring total energy expenditure throughout the distinct time points throughout the trial and knowing their change in body composition, we can determine how many calories they were consuming. And so it's the most accurate way also to measure energy intake if you know the change in body energy stores. So this was an important aspect for the CALERIE trial. So what is doubly labeled water? Liquid gold is how some of us refer to it because it has been hard to obtain sometimes and has been expensive over the years. So essentially water is comprised, as you know, of H2O, and most of the H is in the protium form and most of the oxygen is in the oxygen 16 form. Doubly labeled water essentially has deuterium and oxygen 18. So it's enriched in these. So it's also referred to as heavy water. It has more neutrons, which give it larger mass. So we just dose this orally. So how does this work? And so these two isotopes are what's really important. So this depicts the pool of total body water. And you can think of oxygen and hydrogen going in and out of this pool. And so what this shows is oxygen goes in as molecular oxygen, water, and in all the forms of food, whereas hydrogen is going in the form of water and various food sources. And we then give a dose of doubly labeled water. It mixes with the total body water pool. And then it's excreted over time. And so how is it excreted? Well, after it mixes, you can see that oxygen 18 can come out in the form of CO2 or water, whereas deuterium is only coming out in the form of water. And this can be sweat, urine, fecal water, other metabolic water losses. And by looking at the difference between the elimination rates of those isotopes, we can determine the rate of carbon dioxide production and oxygen consumption. And so essentially we take urine samples, which is the easiest to work with. And we do that before dosing and at distinct time points after dosing. And then we measure those samples by isotope ratio mass spectrometry, and we look at the elimination rates of those. So we're looking at the enrichment in those urine samples, and then we can determine the elimination rates of each of those isotopes. And from that, we can calculate essentially the rate of carbon dioxide, and from the rate of carbon dioxide, we calculate total energy expenditure. And so it's just a very nice metabolic method of determining how many calories is this person burning. And so again, we had that for 4 weeks at baseline so we could prescribe their calories very accurately, or pretty darn accurately. This shows the schedule of assessments. It's not intended to be read, but essentially the point is we did a lot of measurements of all different systems. They had inpatient visits. We had all kinds of physical exams, safety measures, anthropometrics and body composition, metabolic, cardiovascular, immune. We had a lot of questionnaires, muscle function, cardiorespiratory fitness, and banked blood and other tissue samples. And most of the things were done at baseline, 12 and 24 months, but you can see there's a lot of other time points in there. So this is a very intensive study for participants as well as for the investigators and the staff. So here's the consort diagram of what we had in terms of enrollment. So we had 238 eligible participants, 220 were retained and randomized. And then again, it was a 2-to-1 randomization allocation. And so we had, from those 220, we had 145 who were randomized to the intervention, the 25% CR group. And then we had 75 in the ad libitum control group, and 117 completers in the CR group and 71 in the ad libitum group. So pretty respectable retention for a 2-year intervention that was pretty intensive with a lot of assessments. So what did these people look like at baseline? So again, they had to be very healthy. And so these are the two groups. They were well matched. So age around 38, larger percent of women, about 70%, more than 3/4 white. Weights were pretty well matched, so none of these variables differed between groups. But you can see they were mildly overweight on average, so about half of the participants were in the healthy weight range, half were in the overweight range, and they had pretty healthy levels on all of these different classic metrics of cardiometabolic health. Their energy expenditure, which we were really most interested for the CR group, was 2467 kcals per day. So that's how much they were burning. And because they were weight stable, that's how much they were consuming. So when we say, well, we need a 25% CR prescription, their prescribed calorie intake then was 1832 kcals per day. And you could see the range. So what did we find after this 2-year intervention? Well, weight changed as we would hope. And you can see there was a large reduction at 6 months, and then more by 12 months, and then kind of some leveling off and even a little potential backsliding there. And weight loss was not the goal, but it's kind of a result if you're imposing caloric restriction. So just keep that in mind. Although it was not a weight loss study, that is one of the things that should happen if someone's calorically restricted. So the average weight loss was 7.6 kilograms at the 2-year mark, and about 69% of that loss was from fat mass, which is pretty typical in diet-induced weight loss. So what kind of percent CR did we achieve? So remember the goal was 25%. So this is showing percent CR for the 4 time periods baseline to month 6 to 12, 12 to 18, 18 to 24. Well, not surprisingly, we had the highest adherence in the first 6 months, 19.5% CR compared to the 25. But then you can see it was not quite as high thereafter, 9.1% on average. And so the average over the 2 years was about 11.9%. And so again, not really quite at our 25%, not close to it, but so just keep that in mind. Despite that, we did see a lot of other favorable changes. So another thing to keep in mind is there's a lot of variability in the percent CR achieved. So, this is this kind of shows how, some people were more than 25% and some people were really not even achieving calorie restriction. And likewise, in the ad libitum control group, although the average was no weight change, you can see there's quite a bit of variability in terms of their percent CR achieved. So the percent CR means some of these people actually were a bit restricted. So another thing to keep in mind, human trial, yes.
So this is like people kept food diaries, or?
So we did do food diaries at distinct time points, yes. Yes, but this is not based on food diaries. This is based on objective doubly labeled water data. So this is where we had actual, we gave them the dose, we got their urine samples, we measured it, and we had body composition changes by DEXA. And so based on the energy expenditure at each of those time points, the different time points, and this is based on over the 2 years. And then based on those energy change, the body energy stores, so loss of fat mass, loss of fat-free mass, we can calculate an energy equivalent for those. And that's what this percent CR is based on. Yes?
So accordingly, during that time period, is their basal metabolic energy expenditure changed? Did you change their diet?
Great question. Yes, no, we didn't change their diets. Yes, their basal metabolic rate went down. And that's why you would expect some plateauing. You wouldn't expect a linear change in weight because their energy expenditure, their basal energy expenditure goes down. And then similarly, even if they don't change activity, and some people do change, they might do less, they might do more, but the cost of that activity could be less. So if they're doing the same amount of walking, but now they're a smaller mass, then it's not going to cost as much. And so not only does their basal metabolic rate go down as a normal metabolic response to calorie restriction, but their total energy expenditure also would go down because their activity energy expenditure could go down. But no, we don't adjust it because essentially that's one of the principles of CR is that you have this adaptation and then you just maintain that. If we kept titrating down, people would be withering away. But that's a great question. So why didn't we have higher adherence? Well, this is something we don't always like to talk about, but so essentially each person got a very personalized weight graph with their starting weight and the study months, and they had these 3 lines. One was this 50th percentile line, which was kind of the target, and then this lower limit, 10th percentile, and upper limit, 80th percentile. This was considered the zone of adherence, and essentially we wanted people to stay within that range so they weren't losing too much weight, which would be below this line, or not losing enough weight above this line. And so this shows the actual data for one participant. And each person's graph is very interesting and variable. Okay, so that person looks pretty adherent. But this was based on modeling. And so these graphs that were generated for each person was based on a lot of modeling that was done using other data like phase 1 data. And it turns out it was a little bit off the mark. And so what this plot shows, this is showing weight loss relative to study month. And if somebody was actually following this, where they would end up. And so what the blue shows is somebody who achieved 15.2% CR at the 1-year mark and 11.9% at the 2-year mark. And you can see they're in that zone of adherence, but the zone was a little bit off. And so they really needed to be closer down here to be 25% CR. And this graph doesn't even show where they should be for 25% CR at 2 years. And so essentially, it's hard to follow calorie restriction. It's very hard to do that. But if the target is wrong, then that makes it even tougher because you think, oh, I'm on track, and then maybe you're not as much on track. So there are various reasons why we didn't quite achieve 25% CR. But 11.9% is still respectable over 2 years. And despite that, and despite the fact that everybody started out healthy, so they had normal cholesterol, they had normal triglycerides, normal blood pressure. Despite that, we see nice improvements in these things. And this is showing at month 12 in the CR group relative to the ad libitum group, and at month 24. So total cholesterol improved, triglycerides were improved. HOMA-IR is a measure of insulin resistance. It's the homeostasis model assessment of insulin resistance, which is based on glucose and insulin. Those improved. And blood pressure improved, and many other things at both time points. So again, despite them being in the normal range to start, and despite them not achieving the 25%, achieving 11.9%, we saw lots of improvements in those things, in those various markers. And this particular article by James Dorling and others of us is a highlight of all these different systems. And so what it shows is what happened for each of these different systems. So a down arrow means it went down, fasting insulin went down, that's good. Glucose tolerance went up, that's good. So a lot of improvements in cardiometabolic risk factors, circulating hormones, some of them improved, and others, no change, shown by this side-to-side arrow. So some things we did not see a change. Inflammatory markers, oxidative stress markers, immune function, those improved, as shown by all of these different markers. Body composition was generally favorable, but reduction in body weight and fat mass and trunk fat, but there's also a reduction in bone mineral density, which is quite common when weight loss occurs. There was also a loss of fat-free mass or muscle mass, but the lean-to-fat ratio went up. There were metabolic adaptations, so a reduction in mass-specific energy expenditure, but no change in core body temperature, a reduction in sleep, core temperature, cognition, improvement in working memory, but really no change in the others. And this is not surprising because they were relatively young. They were 38 years old on average, and it was not a very long trial, so it's hard to know, if it's having an impact, but we're hoping to learn that soon. And then mental health, again, they had normal mental health. We did extensive screening for that, but we did show an improvement in mood, improvements in quality of life, and dietary restraint. So many favorable changes that did occur in response to 2 years of modest CR. Cardiorespiratory fitness is another really important metric, and this is something that I'm passionate about. And the cardiorespiratory fitness, also referred to as VO2 max, is really considered like the best vital sign because it's really associated with longevity. So higher VO2 max, higher cardiorespiratory fitness, longer life in many studies. Conversely, lower cardiorespiratory fitness, premature death, and greater incidence of cardiovascular disease. And this has been shown in large-scale, long-term epidemiologic studies, both in the US and in other countries. Why isn't this used as a classic vital sign? Because it's difficult to measure. You need a lot of equipment, expertise. It's not something you can just do in the doctor's office easily. And so, but we measured this because we really wanted to know what was going on. And so the most common way to express VO2 max is relative to body weight, so milliliters of oxygen consumption per kilogram body weight per minute. And that's to help normalize for body mass. And so what did we find? Well, we found favorable changes at both 1 year and 2 years, both within the group and in comparison to the ad libitum group. If you express it in absolute terms, however, the absolute liters that someone is consuming during their maximal exercise, that went down slightly. It was significant within and between groups at 1 year. And that's partly because they're a smaller person, they have less muscle, and all of those things. But the interesting thing is when you think of this, well, functionally, which is better? Is the VO2 max in relative terms or in absolute terms? Which is more meaningful? Well, when we think of aging, it's like functionally, how is this person doing? And so we looked at treadmill time because this was a standard protocol. And so if you get to stage, 2, 4, 6, and on, that tells how hard are you working, because it's an incremental test, so either the speed or the grade increases with each stage. So if they're going a shorter time, that would mean maybe their function was compromised. If they're going the same or longer time, it means it wasn't. And so what this shows is they actually went a little bit longer at 1 and especially at 2 years. So they actually reached a higher stage on the test because they're 2-minute stages. So they were going quite a bit either faster or with a higher grade. And so that is a significant functional indication that they were doing better after this intervention. Now, we were very interested in biological aging. And so this is, when we talk about biological aging, it's distinct from chronological aging. So chronological age is how old are you relative to the year you were born? That's every year you gain a year chronologically, that's fixed. But biological age is what is your body's age based on your muscles, your tissues, your physical function, all of those things. And so this has become a very popular and important scientific pursuit to understand more about biological age and how can we impact it favorably. And so this particular metric, the Clemara-Duval method, is based on a whole host of biomarkers here. And so there are many different biological age metrics out there, and some of these biomarkers overlap and some are distinct. But you can see these are all the different things that are going into it. And so this is to look at biological age at these 3 time points over the 2-year period in the 2 groups. And what we found is that the CR group was aging more slowly than the ad libitum group. And so the rate of age was actually biological age was less than 1 year even in the ad libitum group, but it was lower in the CR group. So this is a hint that maybe CR is impacting biological age favorably. Now there's other ways you can look at biological aging as a pace, and so instead of just as one time. So these are 3 different DNA methylation measures. So the other one was based on plasma markers. This is based on DNA methylation. And so there's 3 different metrics here, the pheno-age clock, Grimm-age clock, and Dunedin pace. And the first 2 don't show any difference between the AL and the CR group. No indication that CR is having a benefit there based on these DNA methylation markers. So no effect of CR there, no effect there. But in this one, there was a decrease in the CR group, and relative to AL, it was significantly different. So why these discrepant results? Well, one of the kind of hypotheses is that these 2 clocks are static measures of a single point in time, whereas the Dunedin pace is a pace of aging metric. It's a rate of biological deterioration, or like a speedometer. And the Dunedin pace, which was developed by Dan Belsky at Columbia, is based on a cohort study from Dunedin, and it was 20 years of follow-up with people having 4 measures. So they were born in like 72, 73. They were followed for 20 years, and there's lots of metrics. And so that's thought to be a good measure. There are other ways to measure biological aging, and this is from Nathan Lebressour's group at Mayo in Rochester. These are looking at senescence biomarkers in the plasma, and what this shows is many of them showed improvement in the CR group relative to the ad libitum group at 1 year. Some of them showed improvement at 2 years, and some showed improvement at both 1 and 2 years. So that is a suggestion that that CR may have benefits on senescence markers and senescence proteins. And then this is another analysis this is a heat map from a Senmayo gene set. And so these, again, are senescence markers. And what the heat map shows, and this is a small sample, it's only 8 CR participants, so keep that in mind. But it does show that there's a reduction in those senescence-focused genes. And this was based on adipose tissue samples. That's why the sample is so small, because for the people who provided adipose tissue at baseline, 12 and 24 months, it was a smaller sample. But another marker consistent with some of the plasma-based markers that this may be affecting senescence and actual biological aging. And then there's one other recent study from a colleague at University of Arizona who's looking at glycomic biomarkers. And so this is based on glycosylation, which can change with age adversely. And this is showing that, in fact, there was a reduction or an improvement in the CR group relative to the AL group in these glycomic biomarkers of aging. And so in summary, we found that CR is feasible for 2 years. 15.2% CR was achieved at year 1 or during that first year, and 11.9% over the 2 years. That generally it's safe, so we had a lot of safety metrics. 3 participants were withdrawn, 2 for anemia and 1 because their bone mineral density decreased by 5% or more. So we were very cautious. We didn't want people having adverse effects. We did find that there were many benefits on a lot of the classic markers that you think about in terms of cardiometabolic health, but then also on some of these other ones like biological aging markers, senescence biomarkers, quality of life. We have an article coming out soon looking at diet quality and nutrient adequacy. And so diet quality actually improved and nutrient adequacy was not compromised. We did notice some potentially unfavorable effects, as I mentioned, bone density, bone turnover, so resorption markers were greater than formation, lean body mass, and leg strength expressed in absolute terms. So the conclusion really is that longer-term CR studies should be conducted, and NIA is very interested in continuing this work. So where are we now? And so I want to show you that we had Calorie 1 that was very informative for designing Calorie 2. And Calorie 2 is still ongoing in the sense of analyses. And so one of the studies or one of the grants that is funding continued work with the Calorie 2 data is an R33 awarded to Kim Huffman at Duke. And so this is a network grant where we're promoting the use of the biological samples, so plasma, serum, some adipose tissue, some skeletal muscle biopsy tissue as well, and lots of data. So the data are publicly available, and so really encouraging use of those resources and facilitating a lot more publications and learning more than we already planned to and already have. And so then in terms of where we're going with this, so the network grant is going to be ending in January, and it was resubmitted, and the score was 23, which is a great score. Unfortunately, with things at NIH, it has not been determined whether or not it's going to be funded, so we're preparing a resubmission, which we're cringing at because you can't improve on a 23. Probably can't. Anyway, that we're hoping to be able to continue those activities. So right now, though, we are now 12 to 19 years later, and we're doing the Calorie Legacy Study. And so this started in 2021 and goes through 2026. And this is essentially where we are bringing back these participants from both groups 12 to 19 years later. And it's just observational, but we're doing several different assessments to try to understand what has kind of transpired. So we're getting information about their activity and their dietary habits and other lifestyle things in the interim. But we really want to see, have there been lasting effects? Have there been legacy effects from their participation in this 2-year trial 12 to 19 years ago? So we'll see. We have, we're still in progress with that, and we hope there's going to be some exciting data out of that. We're also using data from the Baltimore Longitudinal Study of Aging, as a comparison group because, there were criticisms which were legitimate that our ad libitum control group is kind of a skewed control group because anybody who was willing to come into this study, do 6 weeks of screening and baseline testing and be randomized to either group for 2 years and come in for all this is not your typical community dwelling person. And we agree with that. And so we really wanted to enrich our control group. And so we have data. We're working with Luigi Ferrucci at NIA and obtaining data on that. And so I want to acknowledge, and this isn't even everybody, but many different people at all the sites, at Pennington, at Tufts, at WashU, at Duke, at NIH, NIA, CALERIE Double Labeled Water Lab, and various funding sources that have supported this work. And I always like to end with a cartoon. Man, some rats have all the luck. Here we are in a nutritional deficiency study, and Herb's doing the effects of alcohol on the libido. So which study do you want to participate in? So thank you very much for your attention. I'll be happy to take any questions, and I think I allowed enough time for questions. Great.
Right, we got some questions.
That was a wonderful talk.
Oh,
Thank you
you.
very much. I had a question about some of the markers. You have modifiable risk markers, and you said they were all normal to start with. But when we study things like this and its effect on aging, maybe we need new markers for what's considered optimal.
Well, and you bring up a really great point. So even though there's a normal range for blood pressure, we know that there's a difference if you're at the lower end, the middle, or the upper end. If you're just scaling, you're still in that healthy range. And whether it's BMI, blood pressure, glucose, lipids, pick your favorite. And so, yes, even though they were in the normal range, it might not have been optimal. And that's why, you can see the blood pressure criteria have changed over time, glucose criteria have changed over time, because we know that what's optimal is not what was thought to be optimal 20 years ago or 30 years ago.
And that brings me to VO2 max. Since VO2 max is considered the gold standard for fitness, might it not, is it adjustable? Have tables been adjustable for weight?
So they are sex-specific and age-specific, and so they're very interesting because I use these in my classes, and so they have to be sex-specific because we know that based on cardiac output, lung volume, all of those things, men have higher VO2 max at every age. I mean, in general, when you take, generalities. But then also we know with age it goes down. That's like inevitable. It goes down with age. And so there's very specific, sex-specific and age-specific standards, but unlike BMI and blood pressure and those other things, there's not like everybody agrees this is the number you should have at this age, or if you're, male or female. So there's different charts, but we do know that it's very modifiable though. And so we know that with exercise training, even in elite athletes, if they train appropriately, they can increase it. And so even though, there's some genetic limits to VO2 max. I mean, some people are never going to achieve the 80-something, 86 or whatever that Lance Armstrong had. But if everybody trains to their maximal capacity, they could improve it. And so we've done exercise training studies with people in their 80s, and you can still improve it. And so that's the great thing is it's modifiable, and your age doesn't hinder your ability to improve your VO2 max. So but the absolute increase is going to be more limited, and it does absolutely go down with age. But it's been just a robust marker across so many different studies, whether it's determined by a bike or a treadmill and different protocols. It's just really, it's a great marker, but not a practical one for clinical use. Yes, Dr.
Yeah, very impressive work. Thank you.
Thank you.
Just two questions. So one, about how these participants achieve these calorie restriction in terms of food intake. Is it reducing the frequency versus the amount on each meal is one. And one related question is the covariates of these calorie restriction. So I know it's impossible to blind them. So one potential covariate is, they're more maybe health aware for that CR group, maybe their behavioral changes, or they just eat a lot of apples, like more fiber, potentially explain all this?
Sure. So, yeah, great question. So at the beginning of the study, I'll show you this. We had, so at the first 27 days, there were 3 9-day diet cycles where we provided the food. And these were the 3 different diets. We provided a Mediterranean diet, a low-fat diet, and a low glycemic load diet. And so these served 2 purposes. One was to ensure if they followed, because we gave it to them, all the meals, that they would be successful with adhering to the calorie restriction goal if they consumed these meals. So they didn't have to think about like, well, how am I going to achieve this? And so it was designed to help them be successful, I guess 3 goals. One, also to kind of teach them this is the appropriate amount. So if you eat this, this is how your body's going to feel, and that's your prescription. That's, your accurate prescription. But then also to offer them some different options, like maybe you prefer a Mediterranean style diet. Do you feel more sated on that, or do you enjoy that? And it has higher fat than the others, or are you kind of the low fat kind of person, or do you want low glycemic load? So we offered these options, and then after those 9 days, of each diet, they didn't have to even think about any of them, but some of them did, and they would, kind of choose to be more like one. We did not impose any particular pattern or composition. But they met with a dietitian regularly, and so there was a lot, and they met with a behavior specialist, a clinical psychologist, regularly. So there were a lot of aspects that the calorie restriction group got that the ad libitum group did not, in terms of, how do you achieve this? And so how they achieved it was very different. And so unfortunately, though, this preceded time-restricted eating. And so I feel like that would have been so helpful. And so back then it was more like reduce, each of these meals. But people did that differently, whether they, reduced more in 1 or 2 meals versus across the board with all 3. It was highly variable, and we didn't mandate that. It was essentially, what's going to enable you to be successful? What can we do? You help us determine what's going to fit with your lifestyle. And this could change over time, depending on what was going on in their lives. And in fact, one participant who was having her kitchen redone, so she couldn't cook, so she was using a lot of prepared meals, and we were providing some foods. She actually, her weight was the best during those. She was right on target because everything was more, precise. Not that we want to encourage people to use packaged meals, but it was, that was a strategy where people could do that if they were having a rough time, whether it was holidays or life events or other things. We would, provide them different tools, either meals out, or, we had what was called a toolbox to try to come up with strategies. Again, very individualized to what's going to help you. If eating more fiber, if modifying this meal but not touching that meal because that's too disruptive with your life. Whatever, is going to work for each person to try to achieve that. And so it makes the analysis very messy, as you point out, because, what time of day were they eating? Did that make a difference? What specific components? I mean, we have done analyses with some of the specific components and other things and diet patterns, but there's a lot of confounders for sure. Yeah, it is messy. The dietary data is always messy, but especially when you're looking at 2 years. And the other limitation is, we had food diaries at distinct time points that we analyzed every 6 months, 6 days every 6 months. That's not really sufficient because they might have been acting differently during those 6 days. And so even though they might have been keeping close track, we didn't use all that data. So it's, there's snapshots. And that's why, when we're looking at percent CR, we're not basing it on their self-report because that's never optimal. We are basing it on doubly labeled water, DEXA-derived changes in body composition.
Hey,
Yes.
2 questions. So since we're in the Evolutionary Medicine Center, I have sort of evolutionary medicine questions.
Sure.
And the heart of it is, I think, the question that a lot of us think about, which is why, if this is so much healthier, why haven't we evolved to prefer that level of calorie intake? But I had heard a long time ago that with calorie restriction, there's a higher susceptibility to infectious disease. So did you measure colds and flus and things like that in your?
We did, yes. And we had different immune function tests too. And so this was adverse events. And so, so adverse events, when you're doing a long trial, you're going to have a lot of them. But these are ones that occurred more often in the CR group than in the AL group. So sure, over 2 years, people are going to have colds, but were they more common? So these are the things that were definitely more common in the CR group. And so sinus congestion is on there, but there's nothing else. Oh, influenza-like illness. There you go. Yeah, right there. So it's just a little bit higher, 3.5 versus 2.3.
Those
But
are years 1 and 2, those two different bars?
oh, yes. Oh, sorry. This is, yeah, these are only in Europe. Sorry. Yes, 1 and 2 years. So this is 3.5 at year 1, 2.3 at year 2. Percent of participants reporting these events, where it was by at least 5% of the sample there. Yeah. And so, and this is where, working with a dietitian is really important. So the other thing is we did provide a multivitamin mineral supplement and vitamin D. And, not that that's like the answer to everything, but anytime you're restricting calories for long term, it's a good idea to just provide some of those things just to avoid deficiencies. But certainly, that's where diet quality matters, and that's why I was very interested in looking at diet quality, because certainly if the calories are low and the quality is low, if they're just, cutting out some of the better things and eating whatever just to stick to their prescription, that's not ideal, and that's where it's definitely more susceptible to infection, to infectious diseases, those types of things, or viruses and that type of thing. And so.
And then my other question is really for the follow-up where you've got the 19 plus years or whatever, 12 to 19 years. Are you going to look at reproductive output, number of children?
I think we do have that on our questionnaire. Well, so, and interestingly, some of the reasons that people dropped out, but this was in both groups, was due to pregnancy. Or, we had to withdraw them, I should say, was due to pregnancy. So that's, but yeah, that's a good question. Yes.
Great talk. I know
Thank
you
you.
mentioned that a lot of participants might have been not like quote unquote normal community members, but in the screening process, did you take into account at all like variation in socioeconomic status to maybe account for lower quality diets in earlier life before this?
And so interestingly, we did have some difference in SES across sites, and particularly so we had a little bit higher, more educated group at WashU and Tufts, whereas Pennington has like these professional subjects where they, there are people in the community who do studies. They treat it like a job. So at first I was like, that's a little interesting. But then it's like, wow, no, they take it seriously. They show up because they're getting paid to do this study and it's their job. SES was looked at in some of the different studies, and absolutely that could affect diet quality. It didn't seem to influence things, but it's a great point because absolutely that could potentially be a problem. But we didn't exclude anybody based on SES if they self-excluded. If it was just like, I can't possibly come in for all these visits, that probably skewed things a little bit. So yeah, and when I say not normal community members, in terms of representative of the community, just because they, anybody who does these types of studies that are just so intensive with these inpatient, multiple outpatient, having to do recording at home and wearing devices and all the blood and then biopsy samples and being willing to be randomized to the intervention. So, but I mean, yeah, they were still normal working adults or, whatever kind of adults from the community. But it's a great point because we know that certainly could impact if they just don't have the means to have a healthy diet. We certainly wouldn't want to make somebody's nutrient status worse because of that.
Thanks.
Tai Chi?
I get to ask a question now. Taichi, I'll come to you in a second. All right. It's really cool. I have two questions. The first one is maybe more difficult to answer, but I've talked to Dan Belsky about this a little bit before, but like a biomarker that captures sort of like, relative change, right? How fast you're changing the speedometer versus the odometer, how many miles you've gone, right?
Yeah.
It's unclear to me how, if something is slowing down the speedometer, especially within these longitudinal studies, it's not also affecting the odometer.
Yeah.
Right?
Yeah, absolutely. And, the other markers are based on a lot of, large scale good data. So, they're each a little bit different, but they are based on a lot of, large sample sizes and DNA methylation and various other metrics. And so, yeah, it's a hypothesis, but I'm not sure. I agree. Like,
I'm more convinced with all the other markers that you show change, right, with respect to caloric restriction than, it speaks to maybe the predictive accuracy, in terms of precision medicine or precision geroscience of using things like, DNA methylation marker of biological age as your one sort of outcome, right? I'm a molecular biologist, so it's
Yeah.
not like I'm taking digs at that.
Yeah,
It's
no,
something
no,
that I
it's-
use, right?
yeah.
But I think my main question is, you have a relatively, broad age range in
Yes.
this. How does caloric restriction, and its impact on aging biology, health and well-being, vary across the lifespan, at least within, as much as you can say within this?
Yeah, because it's really interesting. In some of the Drosophila studies, there was a study by Meyer and I think 2013 in Science of the age at which you start calorie restriction. And if you start it early in life, it's great. But if you start it later, it also has benefits. But then the flip is also true, that if you stop it in midlife after getting the benefits early in life, you're undoing those benefits that you had. And so we don't know in humans. And the reason for the age group, because in the first one, we at WashU we're doing adults 50 to 60, which we considered young because our studies were generally, people 60, 70, 80s. And so we considered that young, but the other studies were like, no, we want to try to get this earlier because you may miss the opportunity to have as much impact. And so the rationale for the 21 to 50 was that's kind of comparable to the age in the animal studies at which, the post-weaning and after, growth theoretically is finished, growth and maturation, so you're not stunting it. And so that was the rationale. But yes, in a 21-year-old versus a 49-year-old, would you expect there to be differences? Probably. And that's what we just don't know.
And that the age at sort of onset, right, was of the caloric restriction was one of the big differences that was kind of a snafu between the Wisconsin and NIA.
Yes, there were a lot of differences, yeah. And so in the rhesus monkey studies, the two colonies, one at Wisconsin, one at NIA, they had some very different results. And there were some very big differences in the study designs, everything from diet quality to age at onset to their genetic background, and yes. Yes, Tai Chi.
Very quick evolutionary medicine question. You mentioned genetic background, but do you see any race-specific effects on that?
Unfortunately, we don't have enough diversity to test that. And so, we had 77% white on average, but then the numbers that were black or mixed race or Asian, and it was relatively small groups, and Hispanic, relatively small numbers for us to be able to identify that, unfortunately. Yes.
I live at Mirabella, which is the retirement
wonderful.
community.
Yes. Oh, yes.
On campus. And we're the group that's most desperate to find out results of this. And I'm sure that a lot of people will extrapolate that this was a study related to us, but it was in a lower age group. And so I think your comments are very important to us, that this group was studied may not be us. And also that goes to the issue of these were generally overweight patients.
Half were normal weight, half were overweight,
okay.
but mildly.
And did you see the ones who were normal weight get the same benefits as those who were
So that's an
overweight?
interesting question. For most things, weight didn't seem to matter, or the amount of weight loss didn't seem to matter, but then in some of the things, it was more about those who were at least 10% calorie restricted might have had different benefits than those who were less than 10%, but less about where they started. But yeah, you could theorize if you're already starting normal, how much improvement are you going to get from there? But yeah, it was stratified based on BMI cut point at baseline as well as age and site, and so the randomization was stratified to help account for that. But yeah, great questions.
Awesome. Well, thank you. Let's thank Dr. Racette for a wonderful talk.